Webinar titled Small Mobile Stem Cells, a Novel Paradigm Enabling Targeted Regeneration of Diverse Human Tissues. Before we start, let me go over a couple of things, please. This Zoom will last approximately 30 to 45 minutes, followed by a Q&A session. For the best audio experience, please keep your microphones and cameras turned off throughout the presentation. Today, I'm pleased to announce our distinguished speaker, Dr. Rahmo. Dr. Rahmo is a leading researcher in regenerative medicine whose work focuses on development and clinical translation of innovative stem cell-based technologies. His contributions have advanced our understanding of cellular repair mechanisms and continue to influence emerging therapeutic approaches across multiple medical disciplines. We also extend our gratitude to the American Academy of Stem Cell Physicians for supporting this educational event and making this webinar possible. Without further delay, it's my pleasure to turn the presentation over to Dr. Rahmo. Thank you, Dr. Rahmo.
Thank you very much, Cynthia, for this opportunity. I visited also for the second time the conference in Florida. I enjoyed the interaction over there too, and met some of the physicians and scientists over there. So it's another avenue for presenting, of course, our technology. And I'm sure direct interactions like this will generate a lot of new ideas and potential applications. I do believe in the interaction between scientists and physicians, because it keeps the scientists, and I am a scientist, more productive and more focused on the target. And I'm going to talk today about multi-targeting using small mobile stem cells. We do think that this is a new paradigm because small mobile stem cells have not only very different characteristics than other cells in general, and in particular stem cells, they also have a different mechanism of action that is unique to them. And I'm going to go through that during the presentation. It gives us an opportunity to present a new mechanism, a natural native mechanism for regeneration that has been not untapped really so far. So we have this great privilege of tapping into a new mechanism and adapting it to medical use. Today, I'm going to talk about two very different tissues to emphasize this platform nature of these cells. So I'm going to talk about regeneration of lung and also joints. And we all know how different these two tissues are in terms of cellular constituency and requirements for the regeneration. So a single agent will be able to perform this regeneration, and that single agent is the small mobile stem cell. I'm not going to delve too much on that, as we all know how important COPD is. This is a disease that affects 400 million people worldwide. It's a completely unmet need. It's a progressive, deadly disease. But we have also other type of chronic diseases like the disc disease and degenerative disc disease and the degenerative knee disease, for example. And we're talking here also hundreds of millions of people affected by this disease. And the solution for that are really not satisfying. There is a lot of room for improvement there too. So when I want to talk about COPD, then I need to mention that this is really a complex disease, but it is mainly chronic bronchitis and, of course, the damage to alveoli, which is a lung emphysema. These are the main characteristics of that complex disease. And we wanted to concentrate on regeneration of the air sacs. These air sacs are really tiny, intricate structures composed of multiple cells. The main important cells are alveolar type 1, which are covering 95% of alveolar surface, and they are responsible for the gas exchange inside the lung. There is also another very important type of cell, which is alveolar type 2 cells, and these cells are responsible for extremely important events, including regenerating type 1. So they are able to differentiate as progenitor cells. They are able to differentiate into alveolar type 1 and reproduce themselves. So one would like to know whether one can influence these cells, and they were our early target for the interaction with SMS cells. There are other very important cells like mesenchymal stem cells, for example, like fibroblast, macrophages, and others. Some of these are definitely also a legitimate target for any attempt for regeneration. In the case of cartilage degeneration in the joints, here we see a very different picture, a very different tissue. This tissue is occupied by a central cell, which is chondrocytes that produce the cartilage. We have different fibers and hypoxic environment. We don't have any blood vessels inside, nor innervations. When there is degeneration, strong structural changes occur, and then we do have innervations and blood vessels coming in. And in this case, that degenerative state is actually um, uh, uh, is affected by the fact that blood vessels are coming in. These blood vessels could increase the inflammation and damage further the tissue. So unlike, for example, the lung, where regenerating capillaries is very important, here in this case, regenerating capillaries and enhancing capillarization is actually something bad and will cause a further inflammation. So we were wondering at the beginning whether the same agent or a stem cell that will increase angiogenesis would actually support regeneration of the joints, like in the knee and the disc. And we were surprised, in fact, that this is the fact that we can enhance regeneration in this very different environment. And I will explain later how that is managed by small mobile stem cells. So what are small mobile stem cells? These are tiny cells. We're talking about 4 to 5 micrometer in diameter, very translucent, by the way, and very motile. They can hardly be seen through microscopy. They are also extremely difficult to stain. which explains also why people didn't see them for all that time. So when we discovered these cells, I mean, the assumption was that these are contaminations and they don't fit anything that is known. So we just examined them for making sure that this contamination is not something serious. And I continued doing that examination for for quite a while. It took me 1.5 years of research to make sure that and convince myself that these cells are really human cells and they are very important for the tissue regeneration. So the cells are accessible from cord blood or from peripheral blood, so we can isolate them. The isolation process takes quite a bit of time, actually months, because these cells come at small numbers. And we use a lot of these distinctive features and characteristics of the cells to do the isolation. And we patented that approach. We had obtained so far 7 worldwide patents, and we're filing for more. So these can be, and that's very important, they can be cultured and then grown to large extents. In fact, we can have more than 27 passages of these cells without any change in potency. And that is the basis actually of our allogenic cell therapy. The fact that we can produce huge quantities of these cells. Now, the cells, although resilient, very resilient to the environment, they are extremely responsive also to inductive medium conditions. So many of these molecules that are responsible for the development of tissue influence small mobile stem cells. And that's completely consistent with regenerative function, because the cells need to sense their environment correctly and do the right response and present the right feedback. One of the most important feature and really unique to these cells is their strong selective interaction with cells. And not any cells, but really cells that are key to tissue repair and regeneration, such as mesenchymal stem cells. So they bind strongly to mesenchymal stem cells. They actually stimulate their proliferation and change their gene expression. That occurs also with fibroblasts, alveolar type 2 cells, and chondrocytes and other cells that we are studying currently. The other feature that is important too, and that is part of what we aspire to establish, what we aspire to for establishing in an industry like approach, is their stability. So the cells are so stable to adverse conditions like pH changes, temperature changes, osmolarity, and chemical attack, that allowed us to create always the same quality of the cells. Just an example of how stable those cells are, they can survive refrigerated temperature for 26 days. Here is an example of what these special characteristics present with these cells. So although the cells are very small, they are able to produce large quantities of extracellular matrix. And we know that all tissue are composed of cells and then the acellular, extracellular matrix. Now, extracellular matrix coming from stem cells ought to be unique, because it has a different function than the ECM derived from adult cells. So we studied that ECM, and in one of our studies, we really observed that the cells are able to differentiate endothelial cells into small tubuli, almost similar to what we see also with extracellular matrix derived from, for example, tumor cells. But in this case, instead of apoptosis occurring for endothelial cells after a few days, we've seen those tubuli stable over a month, actually. And after 10 days, they form also large vessel-like structure. These organoids are quite stable, and we can see one here forming. They are attached to the surface of the culture plate, and after several hours, they actually detach from the surface, and they remain as hanging vessel. And that hanging vessel can be actually seen with the naked eye. So these are large vessels that have been formed through the ECM derived from SMS cells. And that's one of the unique features of the many unique features of SMS cells. So here we see the SMS cells stained actually with the green fluorescent binding to mesenchymal stem cells. Now, you all know how important mesenchymal stem cells. So having a cell binding that strong to mesenchymal stem cells and stimulating its proliferation is immediately meaningful, for us all. In fact, when you detach the mesenchymal stem cells from the cell culture plate, the SMS cell remain bound to these cells. This is how strong this relationship between the two cells is. And this is, as I mentioned before, this is not just binding, but it is and stimulation of proliferation. It is also gene expression changes that are very important for the effect on regeneration. Here is just to emphasize the point using flow cytometer that indeed after detaching the mesenchymal stem cells, SMS cells remain attached to it. And here we see a video, the SMS cells green stained coming out from the glass capillary here. And once they come out, they attach to mesenchymal stem cells that are attached to the cell culture plate. They actually wander with the mesenchymal stem cells. And that attachment is quite meaningful. We see that the mesenchymal stem cells are happily dividing over there. In fact, as I mentioned, they are stimulated in this case. And we tested that on mesenchymal stem cells derived from different tissues. In this particular case, we have the lung mesenchymal stem cells, but there are other also mesenchymal stem cells that exhibit the same characteristics in terms of binding to SMS cells. Now, what we have here is definitely an endogenous cell therapy, and I want to emphasize that SMS cells is not intended, we don't intend with SMS cells to do a differentiation of these allogenic cells into tissue of the host. Indeed, what we want is to stimulate the endogenous cells of the host. And by this stimulation, we would regenerate tissue that is completely compatible with the host. So this is a patented regenerative platform technology promoting cell and tissue regeneration. In this case, we have the SMS cells interacting with alveolar type 2 cells. And what we've seen here is when we remove the serum from the proliferating alveolar type 2 cells, we see a great inhibition of proliferation. We can overcome that by adding SMS cells that re-stimulate the proliferation of alveolar type 2 cells. So they replace actually the effect of serum in this cell culture. And again, here, it's not just the changes that occur to proliferation that are relevant, but there is also a great effect on the gene activity. 1,310 genes are affected in this case. And when we take the 40 most stimulated and inhibited genes, we see a story that really rhymes in with regeneration, and it includes lowering inflammation, reducing apoptosis, enhancing pathogen protection, promoting angiogenesis, and reducing vessel permeability, lowering blood pressure, which we didn't know what the meaning of that was, but it is related to COPD, and inhibiting fibrosis, extremely important. You know, you want to make sure when you select a factor that promotes regeneration, that you inhibit the pathway, the easy pathway to fibrosis. And when we looked at the interaction between chondrocytes and SMS cells, and we were happy to see that more than 4,000 genes were actually affected in their activity there. And if you look to the left here, you see the control for the gene activity of chondrocytes compared to the one after adding SMS cells. The difference is quite phenomenal. So we have here red activated genes, and here then becoming black, meaning no more activation. Green that are inhibited becoming red, and those are from inhibited to activated. So it was quite amazing by itself. But when we looked into the details, we found many items. One of them is quite important. There is an inhibition of matrix degeneration and IL-1, which is a major promoter of inflammation. And that happens through different pathways, not just one pathway, different pathways. We have also inhibition of ossification, which is very important in cartilage repair, because that's what we see with the degeneration of cartilage is the ossification. And very interesting, also an inhibition of vascularization. So through the action of SMS cells, through interaction with chondrocytes, there is actually a contrary effect to angiogenesis. There is an inhibition of vascularization. So contrary to what we see in the case of interaction with alveolar type 2 cells, where we see promotion of angiogenesis and vascularization, here we see inhibition of vascularization. And not only this, when we studied all the inflammatory pathways related to chondrocytes, every one of them was inhibited. So there is a strong inhibition again through many pathways. And in addition to that, there is inhibition of neuronal stimulatory pathways. So, and that results in inhibition of pain. And as you all know, people go to the physician because they have pain. So pain in the joints is very important. So the ability to inhibit that pain and effectively and strongly is extremely important as a physician for the physicians and for the patients involved. If we want to go more and dive into the mechanism at the cellular scale, what we find is quite interesting. Now, we know for the chondrocytes that a signaling pathway like SOX9 is responsible for promotion of proliferating chondrocytes, for the differentiation of mesenchymal stem cells into proliferating healthy chondrocytes. And that inhibits further differentiation into the pre-hypertrophic chondrocytes and the hypertrophic chondrocytes. These are differentiations you want to avoid because they lead either to apoptosis or endochondrial ossification. So that's the ossification process. This process is actually stimulated by RANKS2, which is a signaling molecule inside the cells. So ideally, you would like SOX9 being stimulated and RANKS2 being inhibited. So what does the SMS cell interaction with chondrocyte do? In fact, this is exactly what SMS cells do. They stimulate SOX9 not by just one pathway, but by two pathways. And they inhibit RANKS2 by three pathways so that they ensure that proliferating chondrocytes are made and inhibited from the ossification pathway. And there is no drug in the world that can achieve that, really. This is a smart drug, the smart drug SMS cells that is able to do that. Now, when we compare small mobile stem cells with other stem cells, definitely the fact that we have these cells from adult tissue is important. It gives a layer of safety that we are used to from adult stem cells. But at the same time, it does not miss on the advantage of embryonic stem cells and iPSC, which is mainly the ability to produce large numbers of these cells. And beyond that, there is also this resilience and the small size, which led us to figuring out a new approach on accessing the lung, which is a nebulization. So indeed, these cells are small enough, resilient enough to be nebulized without any effect on their viability. Resilience also helps us make sure that these cells survive an hypoxic environment, like, for example, the intra-articular space. So cell therapy is definitely something that is a cornerstone now of advanced therapy. However, it is facing a lot of challenges, be it safety, potency, scalability, targeting, engraftment, and survival. Fortunately, all these items are really not a problem for SMS cells. So if I would like to describe potency, there is very strong ground for strong potency at multiple levels. Cell biomanufacturing is highly scalable. There is a strong selective and direct binding, which ensures good engraftment. And the cell administration can be directly to the lung, non-invasively. These cells are resilient, small, and with low immunogenicity. Only this low immunogenicity, really extra low immunogenicity, ensures that we can use these cells without any immunosuppressant allogenic. And when we tested these cells on rats that had that had been damaged in their lung using a special enzyme, elastase. We saw great results, in really less than 10 days. Two application was able to recover the alveoli by 100%, which is quite amazing, given the scale at the time. This is just an early experiment that we did on rats, and we tested the functional recovery too by using a treadmill, mimicking actually what we do with patients on a 6 minutes walk test. We wouldn't have the patient running on a treadmill, of course, but this is the best comparison. So, as I mentioned, the cells were actually able to uh, 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 nebulize, and their size, 4 to 5 micrometers, actually a fit with the droplets that can reach the periphery of the lung. And this is what we're doing right now. We are nebulizing the cells using a mesh nebulizer and applying it directly to the lung. We expect a stimulation of alveolar type 2 proliferation and regeneration of type 1, modifying the activity of many genes, and stimulating mesenchymal stem cell proliferation and stimulation of capillary formation. So regeneration is always complex. It takes a lot of agents to make it happen. And having this multi-targeting using a smart drug like a smart cell make absolute sense. Here, this is our clinical trial on the COPD, which is being run now in Australia. We have 18 mild to moderate COPD patients as requested by the FDA. And we finished the first cohort now with a low number of SMS cells, and we're moving now to the medium number of SMS cells. And then at the end, with a high number of SMS cells. But we already seen with a small number of SMS cells, very clear signs of efficacy, and this is quite promising. But that's, of course, not going to hold us from attempting to test these cells on many other indications. We are interested now, not just in COPD, but also in lung fibrosis. We're interested in orthopedic wound healing, also small vessel disease, cardiovascular systems, and also in neurologic diseases. We think that the fact that these cells are inside the blood, circulating the whole body, means that they could be instrumental to several organs for the regeneration and repair of several types of tissues. And indeed, this is what appears to be the case. And we have a long road ahead of us on trying to understand all the potential of these incredible cells, really. We tested the cells on other subject matters, on domestic animals. And when we do that, we actually use the human cells all the time. And here we see, for example, the effect of SMS cells on the torn tendon. In less than a month, this torn tendon in a horse was completely regenerated, which is quite amazing. Here in this case, this dog was actually not treated localized, but he had an IV infusion, and we were treating disc degeneration, but it turns out that the dog had a a wound that was non-healing and went all the way to the bone, and the dog risked losing his limb. And after 1 week, we were informed that this wound actually started to granulate, and it is completely closed after that. So even these IV infusion are able to provide for the targeting and the homing of the cells into the right places. We produce these cells in our biomanufacturing site, CGMP biomanufacturing site in Sorrento Valley. We have dedicated a lot of time in perfecting our biomanufacturing scheme. So these cells are produced in high quality. We've been using it also for human, of course, not just for COPD, but also for other indications. And that's all I got for today. So I would like to thank, of course, the distinguished researchers who worked with us continuously. I want to mention also Dr. Roger Schechter, who is our medical director, Dr. Jason Kirkness, he is our chief clinical officer. We have also great advisors like Professor Peter Wagner, Professor Rasan Kassab, James Shirley, and Gary Seek, and Richard Chucks. And I think we should be proud of being associated with these people. Thank you very much.